in the process of going live.
Yeah, we're live now. Go ahead, Graham.
Hello and welcome to another episode of the BiotensegriTea Tea Party, organised by the Stephen M. Levin Biotensegrity Archive. And today we're going to be listening to Dr. Gregory Morgan talking about history of virology and how it's applied to tensegrity, where he came across it many years ago, come across the link. So my name's Graham Scarr. I'm coming from Nottingham in sunny UK. And I'm joined by my co-host, Chris Marita Clancy. Hello, Chris.
Hi, Graham, and hi, everybody. I'm Chris Marita Clancy, coming to you from Coast Salish territory, very close to Vancouver, BC in Canada.
And my other co-host, Susan Lowell de Solorzano. Hello, Susan.
Hey, Graham. Hey, everybody. Thanks for being here. And thanks, Dr. Gregory Morgan, for being here. We're really excited. I'm coming from Washington, D.C.
So what we always do is we have a toast to start proceedings. So take a glass or cup of your favourite tipple. Let's host a toast to biotensegrity, virology, and long may our studies of biology continue. Cheers.
Cheers.
Okay, and I'll turn it over to you, Susan.
All right, great. I'm going to spotlight Dr. Morgan. And can you tell us, sorry, I'm trying to do two things at once. Dr. Morgan, thank you for being here. You're coming in from New Jersey?
Yes. Thank you so much for having me and inviting me to your group. Yeah, I'm a professor at Stevens Institute of...
Wait a second.
Sorry, I pressed the wrong button.
Oh, that's okay. We accidentally muted you. Greg,
Okay.
tell us a little about yourself.
Sure. Well, as you can probably hear from my accent, I'm actually a New Zealander by birth, and I'm now a professor at Stevens Institute of Technology in Hoboken, New Jersey. I work in the history and philosophy of science. So particularly biology is where I've sort of specialized. And for a number of years, I've looked at the history of virology and some of the sort of more philosophical implications of discoveries in virology. So anything else you'd like to know?
Yeah, I mean, I know you've got a great presentation that I'm really looking forward to, but how did you get to where you are? This is a rather unusual position, that you've found yourself in. Not too many people doing history of science. It's really wonderful. How did you get there?
Right. Yeah, as I said, I grew up on a ranch, a sheep and cattle ranch. So I've always been interested in biology and animals and plants. But at a certain point in time, I decided I didn't want to be a farmer or a rancher, and I thought that universities looked great. So I was at university when I made that decision. I ended up actually being an exchange student at University of California at Berkeley for a year, and I totally loved it and was thinking about what I wanted to do. I was actually in Powell's bookstore in Oregon, and I came across a book on the philosophy of science. And I thought, wow, I didn't even know this field existed. And it seemed like it asked all the interesting conceptual questions about science without actually having to be in a laboratory. So I kind of switched and became a graduate student and finished my PhD at Johns Hopkins and then went into the academic job market and ended up here in New Jersey.
It's so cool that you find a book and it's like just the field you didn't even know existed that you were destined for.
Oh, yeah.
How did you get into virology? And
there's a sort of a field called philosophy of science, and one of the centers for that is at the University of Pittsburgh. It's got a very strong concentration of scholars there. And so they encourage people to specialize in a science as well as a philosophy in order to be good at this sort of interdisciplinary thing. So I was encouraged to get a master's.
Zoom that I'm just putting on so that I can.
Sorry, We had
No, that's
somebody
okay.
that needed muting. Go ahead.
Yeah, so I was at the University of Pittsburgh and I was told, well, you need to know some more biology. So I ended up in a virology laboratory there studying bacteriophages, which are the viruses that attack bacteria. And I've kind of fell in love with them. They're a very elegant organism, very simple life cycle, and they seem like a good system to try and understand life.
Oh, that's great. Well, I don't want to keep anybody from your presentation. So
Okay,
I will disappear for a few and turn it over to you.
okay, great.
All right, Dr. Gregory Morgan, everyone.
Okay, let's see here. So, can you see that?
Yes.
Okay. So I want to talk today a little bit about sort of the history of virology that leads up to the really interesting time in the 50s and 60s when we start understanding what viruses are made out of and how they're structured. For a long time, there's sort of two different types of viruses that we knew about. There were the rod-shaped ones and the spherical ones. And the rod-shaped one, the most studied rod-shaped virus was tobacco mosaic virus, TMV. And a number of very eminent scientists studied TMV, including Maurice Wilkins, Jim Watson, Don Caspar, Rosalind Franklin, and Aaron Klug. And I'm going to mention them a little bit more. And then another virus that was very important, another plant virus, was tomato bushy stunt virus. That was also studied by Caspar and Klug. So TMV, or tobacco mosaic virus, it's sort of a model for virus assembly. It has a helical structure like DNA, and it's the most well-studied virus. I'm going to talk a little bit about what happened before the 1950s. So TMV, if a plant gets infected by this virus, it gets this sort of mosaic pattern on its leaves, and that's why it gets called tobacco mosaic virus. The first virus that was discovered in the late 19th century, and important because of the economic value of tobacco. What was really interesting is that in the 1930s, we could see that we could actually crystallize viruses, which was surprising to some people because if viruses are living things, it seems strange to be able to crystallize them. But Wendell Stanley, who was a famous Nobel Prize winning scientist at Berkeley, crystallized them and got famous for that. This meant that you could do X-ray crystallography of the crystals and try and learn something about the viruses themselves. Also, there was the rise of electron microscopy in the 20th century, where here's an image from the 1940s, where we're finally able to see what the virus looks like. It's a little murky there, and you can see it's hard to know what to infer from these early micrographs. But a natural thing to do is if you can crystallize a chemical or a virus is to put it in an X-ray beam and then analyze how the X-rays diffract through the crystal. And this was done by Bernal and Van Keuken in the early 1940s. Van Keuken is going to be an important figure for mentoring Don Caspar, who's going to be the key player later on. So in the 40s, people speculated what the virus looked like to try and explain those micrographs that you saw earlier. This was a German virologist called Schramm who thought, well, the virus, maybe it's shaped kind of like rods stacked together. And that would explain the different lengths that you could see, but it's still very basic. That's Schramm there in the top left-hand corner. There were other people that were working on this. There's Wendell Stanley down there in the bottom right, who was the one that did the crystallization, and one of his colleagues, Frankel Conrad, who actually supplies crystals to various crystallographers, including Rosalind Franklin. So I like to think of the 1950s as the decade of the helix, because really important helical structures were discussed, including DNA, as I'm sure you'll know. TMV turns out to be helical as well, and the alpha helix in proteins, all major discoveries of the 1950s. This is Maurice Wilkins. He was a New Zealand British crystallographer who was working on tobacco mosaic virus. He was actually working with Rosalind Franklin as well, if you know the discovery of DNA structure story. And so they published something in 1950 where they had significant enough results to submit to Nature, the preeminent journal of the time. And you probably know the well-known story of Crick and Watson, who are in Cambridge here in this photograph. They're going to be important figures in this story too, which is sort of goes beyond the DNA story that most people know. A lot lesser known is the virus story. One thing Watson was working on around about the same time he was thinking about DNA was TMV structure. He thought that he could get insight into nucleic acids by looking at viruses. And at one point in time when he was looking at DNA structure, he was told he couldn't compete with the King's group where Rosalind Franklin was working. So he worked on TMV, which has RNA instead of DNA in it. So he thought that maybe he could learn something about the structure of RNA. This is what his diffraction photographs look like. You can see there's structure there and that there's definable dots, but it's still not particularly elegant, especially if you see Rosalind Franklin's work on the same thing. So Don Caspar and Jim Watson speculated on what TMV looked like, and they came up with this structure. They thought that perhaps there's 12 helices of RNA in the center, and then these big bulbous proteins around it protecting the nucleic acid in the middle. And so this was one of their first attempts at what the structure of TMV would look like. This is if any of you have read the book on Rosalind Franklin by Maddox, this is the cover photograph, and this is actually Rosalind Franklin in 1956 at a virus meeting in London at the SIBA Foundation. She also was working on TMV. And as the story goes, right, she experienced a lot of sexism. For example, she wasn't a member of the famous RNA tie club. That's Jim Watson in the right-hand corner and Francis Crick in the top left, who were trying to think about nucleic acids and RNA and DNA and what the relationship
Dr. Morgan,
was. Yes.
I want to interrupt you. The RNA tie club, can you tell us a little about that?
Sure, sorry. Yeah, the RNA tie club was as an informal group of scientists who were interested in kind of the intersection of chemistry and physics and biology and the implications of the new DNA structure. One thing they were very interested in is trying to work out what today we would call the genetic code, which is how is DNA and RNA and proteins related to one another.
And are they?
They made themselves a tie that you can see them wearing there, and they actually named themselves after amino acids to limit membership. But my point I was trying to make with this club is that it was also it's an interesting sort of anecdote, but it's also got another dimension in that Rosalind Franklin wasn't invited, and this was maybe a symbol of some of the sexism at the time.
Thank you for going into that.
Sure. So here's what Rosalind Franklin was working on. Her micrographs, her diffraction photographs of TMV were very detailed and showed a lot of information. Even if you're not a crystallographer, you can see there's certain patterns in the in the photograph there, and the secret was, how do you work backwards from this photograph to infer the structure of TMV? Just to compare it, this is the famous photograph, X-ray photograph of DNA. It was also taken by Rosalind Franklin and used by Watson and Crick when they were constructing their model without her permission. It turns out that the model of TMV that I showed you a few minutes ago that had the RNA in the center was incorrect, and that RNA actually was intertwined with the proteins at a radius of 40 angstroms. So you can see the big peak in this graph here is RNA, which made it harder to work out what the structure of RNA was, but it was sort of a big advance on thinking about how proteins and RNA interacted with one another. I just mentioned this in part because that Rosalind Franklin and Jim Watson were sort of considered themselves friends even after the discovery of DNA. And if you look at the second paragraph of this letter, this is a letter from Jim Watson to Stanley at Berkeley saying, Rosalind Franklin's X-ray analysis of TMV is becoming more and more exciting. So he was aware that she was sort of on the cutting edge of TMV research, and they interacted with one another, and they talked about science a lot. And Rosalind Franklin was very friendly with the Cricks. Another person that comes into this picture, the story, is Aaron Klug. This is the man in the glasses on the right here. He's a South African physicist, mathematician, biologist. He would later become the president of the Royal Society and win a Nobel Prize. But he initially started his career in biology by working under Rosalind Franklin. So he was sort of Rosalind Franklin's most important student at the time, or most important collaborator, I guess. He had a PhD at that point. So the picture of what TMV looked like was becoming more detailed, and the thought was that perhaps it was a little bit like a spiral staircase, where each unit of protein could kind of be added to a stack of proteins, and so it could self-assemble into its shape. This particular image doesn't show where the RNA is, which is sort of in the middle of it, in the middle of each step at a certain radius. Here's another letter here to Rosalind Franklin from Jim Watson, sort of illustrating that they actually interacted and were colleagues, and that there wasn't fallout from the DNA race that ended their relationship or anything like that. So by 1956, these people were attending crystallography conventions, conferences to talk about virus structure. Here you can see the tallest man on the left is Francis Crick. Right next to him is Don Caspar wearing the sunglasses and the mustache. Then Aaron Klug in the middle, and then Rosalind Franklin, and then another couple of very eminent biologists, John Kendrew and his wife. Crick and Crick and Franklin interacted. Here's a letter here from Rosalind Franklin to Crick, where they're talking about the structure of TMV, getting right into the sort of weeds about how tight a helix it is, how many units per turn, and how to interpret the different spots, or they call them layer lines, on their X-ray photographs. I took this quote from Watson's book, Genes, Girls, and Gamow, where he says, in the mornings and many evenings, I was in the Cavendish taking X-rays of RNA containing potato virus X. It was my response to an overnight visit from Rosalind Franklin, who stayed with the Cricks. Listening to her treat Don and me as insignificant players in TMV research, I felt the need for another plant virus to call my own. So Watson at least saw himself as in competition with Franklin, even on viruses, sort of mirroring the earlier competition, you might call it, for the DNA structure. An important meeting happened at what's today was the Novartis Foundation. Then it was called the SIBA Foundation. And the earlier photograph of Rosalind Franklin was taken there, and as well, this is Watson and Crick at the time in 1956, talking about virus structure. Again, this is the picture of her at that same meeting. There's not very many pictures of Rosalind Franklin, and this is maybe one of the best ones that exists, I think. Over time, the X-ray diffraction patterns from tobacco mosaic virus got better and better, and more and more information you could glean from them, and you can see this by the number of spots and how much resolution you can get from someone who's a very skilled preparer of specimens, as Rosalind Franklin was. So the structure of TMV then starts looking more and more like what we think of it today, which has a hollow core, and then the RNA is threaded in between the protein subunits, and that's expressed with those black dots up and down this cross section. There was a World's Fair in 1959, and the Cambridge group decided to make a model of TMV and put it in there, and this is their large model. It's like a human-sized model. You can see to the right of this on the picture there, that's a fire extinguisher. So this is a huge model. That's now in Cambridge at the Library of Molecular Biology. And unfortunately, in 57, Rosalind Franklin died of cervical cancer. This is her office and how it looked after she died, taken by one of her students. So now I want to switch to the other type of virus, and maybe the one that's more important for tensegrity, which is spherical viruses. So there's a few that were studied in the early days, 2 plant viruses and also poliovirus. This is some of the first micrographs, the first X-ray diffraction photographs of a crystal of tobacco bushy stunt virus. And I'm just going to stop this share for a second to show you what's happening here. So if you look at a model, this is what sort of the crystal of crystallized viruses would look like, where each of these white things is a virus. And then depending on the axis that you put the X-ray beam, when you irradiate the crystal, it can be in different orientations. If you look at it dead on the front of this one, you can see there's a little 5-sided pentagon there, and that would be in the direction of 5-fold symmetry, which is sort of rare in nature. And so this is what Don Caspar was able to do, was actually show what happens when you look at the crystal of a spherical virus in the 5-fold pattern. So I'll show you again another image here. So this spike with the 5 on it was an illustration of the fact that there was 5-fold symmetry, which was a real surprise for most biologists. It wasn't quite as surprising for Watson and Crick and Don Caspar, because they speculated that icosahedral symmetry was what you might expect. So the thought was that what we call spherical viruses are actually like little soccer balls. They're little icosahedra. And if you look at this diagram, the red vertices are five-fold, axes of five-fold symmetry going from the red dot to the center of the icosahedron. There's also three-fold and two-fold symmetries in this shape too. So sometimes it's called 5-3-2 symmetry. And Watson and Crick published this in 1956, where they looked at different types of symmetry. They thought that perhaps the icosahedral symmetry was the best one because you could have the largest number of subunits. And so it's the most efficient way of enclosing space. And Don Caspar was a bit of an artist as well as a scientist. He drew this diagram to illustrate it. If you look really closely, each of these subunits is actually a hand, a human hand. And the idea was that if something has icosahedral symmetry, you can take 60 subunits and put them in an identical, they can be placed identically and create this icosahedral shape. So all the contacts are the same. Every hand is touching every other hand that's nearest it in the same way. And this was the picture in 1956. It's an elegant picture. This is what they expected all spherical viruses to be like. The problem was that there was now evidence for viruses having more than 60 units. So how do we arrange more than 60 subunits on the surface of a sphere? They can't be identically arranged because the maximum number that you could arrange is 60. Yep. And so here we get closer to what I think a lot of this group may be interested in. The polio work that was done in 58, 59 was actually published in the newspapers. And you can see here they're experimenting with ping pong ball models and other ways of arranging the proteins on icosahedron to try and explain how polio is structured. And then this newspaper article was seen by this man, John McHale. He was a member of the independent group who were sort of cutting edge pop artists in the UK who were trying to revitalize art by bringing in popular culture, but kind of a precursor to what happens in the United States with people like Andy Warhol. Anyway, they were looking for aesthetic inspiration wherever they could find it, and especially in science. So the type of thing that Caspar and Klug were doing with viruses sort of appealed to them. There's one of their exhibitions from the early 50s where you can see they're, trying to find new ways of doing art that blends art and science and looks at a biological illustration. And they were also interested in Buckminster Fuller, as I know a lot of you are. So McHale actually arranged for Buckminster Fuller and Caspar and Klug to meet. And so Buckminster Fuller, on a trip in the UK, spent some time with Aaron Klug, who found him a very stimulating figure. And it turns out that the way Buckminster Fuller's domes are structured solves this problem that they were worried about, which was how do you arrange more than 60 units on a sphere? Since a lot of the geodesic domes have a lot more than 60 structures, they often 60 subunits. They typically have a perfectly spherical one would have a multiple of 60 subunits. And so they were, as a lot of you know, they were used for radar domes and things like that. These are good to look at in part because you can see how some of these arrangements of triangles are arranged in groups of 5 and some are arranged in groups of 6. So they're slightly different. when they're in a 5 or when they're in a group of 6. And this is what interested Casper and Klug. They also, these virologists, like this idea that since you can build a dome with unskilled laborers, as was done in Afghanistan in 1956, that this was a good model for how nature must do it. How does nature assemble a viral shell? It has to be with relatively simple rules, since it's a process that's being done without any sort of conscious oversight. And so they imagined, like, here goes a really simple way of building, where you label the 3 sides of each triangle you're building with 1, 2, 3, and you make a bond between 1 and 2, or a bond between just 3s and 3s. And if you do this, you can show there's one facet of a 12-sided icosahedron there with all the subunits on it. This is in Aaron Klug's handwriting. Okay, so the tensegrity spheres of Kenneth Snelson and Buckminster Fuller also follow these sort of similar rules, but you've got a certain number of subunits that have got to be put together. As you know, the key idea is that you separate tension and compression components, and you create this adaptive structure, and that was sort of attractive because this is what we want viral shells to be too, that are self-assembling and adaptive. On the right is a model I found in Cambridge that was built by the scientists at the time. And a lot of you know a lot about Kenneth Snelson, right, and tensegrity. The things that are interesting for the virologists is that they're in equilibrium. They bounce back from deformation. And I think a lot of you have probably seen the Needle Tower in Washington, D.C., which is inspiring since it's stable and none of the struts touch one another. As it turns out, that Klug thought that what they were influenced, what he called, by a misprison. He had thought that the subunit and Bucky's geodesic domes just had given them so that you could put them in either a group of 5 or a group of 6, when in fact a lot of the domes were pre-calculated in advance. But that didn't hurt the idea that this was still an inspirational analogy for people like Klug and Casper.
Dr. Morgan?
Yes.
Sorry, can you go back? I'm sorry.
Yep.
Can you tell us about that term, misprison? Because
Sure.
yeah.
Sure. Yeah, this is a word that comes out from Harold Bloom. It comes out of literature studies. And it was initially the idea that, say, if you're a poet, you can be inspired by earlier poets, and often by misinterpreting what they meant. So the thought was people can have influence over other people even when they don't understand the person they're being influenced by. So in Klug's case, he thought that he understood how Bucky had built his domes, but he misinterpreted what had happened. But it still was a really important influence for him. I think misprison happens all the time, actually, in cultural change, where people are influenced by earlier people, even when they misinterpret what the earlier people were doing. Does that make sense?
Yeah, really key concept. Thank you so much.
Yeah. Initially, Howard Bloom, who was an important literary thinker, was Don Caspar's friend at Yale. So they were sort of so Caspar is a really interesting scientist in that he's sort of interacting with people outside of science. And Klug too thought that he gave Buckminster Fuller sort of the time of day when some other scientists thought he was a little bit crankish. And he had this sort of esoteric vocabulary that was hard to penetrate. Yeah, so sort of take my hat off to Klug. He was open to other ideas, and they actually made an early a movie together, Klug and Buckminster Fuller, that was one of the first in the Horizon series of documentaries on science in the UK.
And is that accessible that you know of?
I've tried to track it down. I think it's in an archive somewhere. It's a little bit like the PBS science documentaries we would do in the US, but it was done in the UK. It would be a good project to try and track it down. I think it was made around about 1960.
Oh, Bonnie DeVarco put in the chat just now. Yes, it is online Horizons. Thank you,
Ah,
Bonnie.
okay. I couldn't find it when I was doing this 20 years ago, so I guess someone has put it up online. That's great.
Thank you. All right,
Yeah.
back over to you, Dr. Morgan.
Just a few more slides. I'm sorry. Bear with me here.
No, we are loving it.
Okay. So one of the key ideas that comes out of this intersection between tensegrity and thinking about how to put more than 60 subunits together into a spherical arrangement is quasi-equivalence. And so, as I mentioned earlier, one way of seeing it on the dome is that if you look at that picture of a dome, you can see there's a five-fold symmetry circle. It's actually I painted it in a darker color. There's two of them, right, on the left and the right. Actually, can you see my cursor or no? No, you can't, right?
Yeah, no, we can. Yep.
Oh yeah, so right.
There's the pentagon. Yep.
Yeah, there's the pentagon, and if we consider a triangle that's in a pentagon or what they call a pentamer, with one that's in a group of six, there's slightly different angles, and the triangle has a slightly different shape. And so this is the idea that Casper and Klug wanted to transport into proteins, and the idea that you have an identical protein that would have a little bit of give in it, so it could fit in slightly different arrangements. And this was the key to the 1962 paper to understand how you could get different sized viruses built out of the same subunit. Okay, and then Casper and Klug worked together, and they managed to derive this very elegant equation, which limits the number of multiples of 60 that you could put together, and it's called the T number. So only certain T numbers are available, and they're given by this very simple equation. And it turns out that this is one of the very few simple biological laws that we find in biology that look a little bit more like physics. So these are allowable T numbers: T equals 1, T equals 3, T equals 4, T equals 7, T equals 12. And there is no T equals 2 number. You can't build one that's T equals 2. So you multiply the T number by 60 to get the number of subunits in each case. So as you get larger and larger viruses, you get more and more subunits. And so it turns out the tomato bushy stunt virus, which is the one that they were looking at earlier with polio as well, is a T equals 3 structure. And on the right there, you see what a high resolution picture of the virus looks like. And you can see how it maps, every subunit maps onto this geodesic dome. And here's a picture I like this of Casper. When I interviewed a lot of these scientists, Casper was still living at this point when a lot of them have all these models of things that they've built to try to inspire themselves to see the different structures.
Can
So
we go back to that photograph for just a sec?
sure.
I want people to notice in the back, that really big one with another smaller one in the center. So is your understanding that he was also building tensegrities? I don't see any
Yeah,
tensegrities per se.
I think they did build some, but a lot of them, once they'd got this idea of quasi-equivalence,
Yep.
they ran with that idea. So for example,
Yeah.
this one here, this was Caspar's way of doing it. He made the structure out of these little cones, and then the cones could deform a little bit and to be slightly different in different spots around the surface.
So then he could get the hexagonal
Yeah, so some of
connection
it,
as well as the pentagonal
yeah, so
connection.
right here, I think there's a pentagonal one there, right? That's
Nice,
5, and that one's 6. And so, yeah,
nice.
and I think some of this, there was a company called Geodestics. I think might be from that company that made toys that you could buy and build your own models. That was inspired by Buckminster Fuller.
But then we also see one made of, it doesn't look like ping pong balls because, or maybe it is, the globby one that's kind of,
Yeah, I think
yeah,
that's actually more like, looks like a chemical one where the black is carbon and the white is hydrogen or something.
that's
Um,
great.
yeah,
Thank you.
actually, Caspar was my key to get into this whole story. He was a very interesting scientist. Um, he loved to talk, so I went down there and he basically talked to me for 3 days without stopping, trying to convey all the different things about the history.
There's
Um,
probably a book in that, Dr. Moore.
yeah, and he interacted with Buckminster Fuller in the United States too. I think he may
Wow.
have, and so yeah, he's an interesting guy. He had trouble publishing things. He published the 1962 paper with Klug. He had other papers that didn't publish for years. He just kept on modifying them. And some of his early stuff when he's publishing with Rosalind Franklin, she did all the writing. So in conclusion, I was hopefully one thing you can get away from this was that like all this progress in structural virology was driven by theoretical insights from math, art, and architecture, including tensegrity. And I got one other slide here that's going to show you. Unfortunately, this is now out of date because the new Times Square ball isn't like this anymore. But it used to be a T equals 3 structure. So at New Year's Eve, you'd see a T equals 3 structure falling down the pole in Times Square in New York. So that's all the slides I have. Let me end here, and I'm quite happy to take questions, and we can talk about some of this.
Let's see. Get myself up there. Okay. Having trouble turning my video on. Hold on.
Okay.
Wow, that was fantastic.
Hopefully I didn't overwhelm you with too much detail there, but I figured it gives you sort of a picture of how the field developed, and a lot of this is, I mean, that ends in the 1960s, but even today, spherical viruses are still governed by those rules. So the 1962 theoretical paper has turned out to be like a long-lasting contribution to structural virology.
That's great. Steve, did you want to
Yeah,
jump in?
well, one, I want to thank you for that wonderful talk. And I started out playing around with the round ones too. But the problem, of course, is that we get away from the round as we develop an organism, and we had to figure out ways of developing not round tensegrities.
Right.
Now, one of the things that I don't mean to be critical of the researchers at the time, but they were all working with dead viruses. If you do electron microscopy, and to me, that's like anatomists working on dead corpses. You really don't get the vitality of the system. You need that bolt of lightning that the Frankenstein monster got to bring it back to life.
Right.
And what I see is not that round sphere, but a pulsating round sphere that is constantly changing and pulsating. Um, so I think there's more dynamics to it, and I think that's where because in those days soft matter physics and semicrystals were in a topic of conversation. so
Right.
that they didn't really understand that part. And they're working with hard crystals, and I don't think viruses are hard crystals. I think they fit in with other biological substances, which would be soft matter.
Yeah, I think that sounds right to me. That especially if you're looking at trying to infer crystal structure, virus structure from crystals, you're going to get an average structure just by the way the mathematics works. So you won't get the individual variation, and you'll just get the average. Um, and yeah, the next step is, as you say, to try to get the dynamics that involves how to build the virus in the first place out of pieces and how that all come together, how they come apart when they're in a new cell. Yeah, how they merge with the membrane when they infect a cell. So yeah, I imagine if we could see this, they would be jiggling around and doing all sorts
Yeah.
of things.
Well, I have a couple of models that do that, um, that I didn't make, but I picked up that show that you can expand and contract these and change the shape, starting with a round, looking like a round virus. So that, and we of course think in terms of tensegrities more than we do a rigid dome.
Right,
Our things are alive and moving, so they're tensegrities and include the energy dynamics that's within a tensegrity.
right. Yeah,
Yeah.
actually, one of the other people that have contacted me over the years is people that are interested in building structures in space, and the
Yeah,
idea that tensegrity structures would be a way to go.
right.
Um,
That's Vita's son Spiral was the one who was involved with NASA in doing that. Yeah, I
yeah,
knew him. I dealt with him over time. Um, and of course, then you get into all this stuff done on cells done by Ingberg, all the cells doing the same kind of thing. And again, he's primarily working with round things, and as you get into larger organisms, you lose the round.
right. Yeah, I think
You lose,
even at the molecular level, I think you can say something similar to that too, which is the protein subunits that make up biological structures aren't static either, and they can move and flex and change their shape a little bit in different environments, which is what we want, right? We don't want to have life be static.
right. So what we do in tensegrity then is put life into the icosahedron, so it becomes a dynamic tensegrity,
Right.
a dynamic icosahedron.
Yeah, that's a good point.
Oh, that's great. And boy, what a wonderful presentation. Graham, I know you've done a lot of work with structure and model building. Did you have?
I have a question. Um, about 20 years ago, somebody, her name's Twarok, published a paper on viruses with like Penrose tilings, which are
Right.
non-periodic. And I haven't found anything about it since. Did it die a death or?
Yeah, no, I remember that paper. There were there was various attempts to try to, I think, make even a more general theory than Casper and Klug's and derive it from either thermodynamics or yeah, something more elaborate tiling theory. I'm not sure what impact it had on the field. Part of the reason is that maybe this was a unique period in time because there wasn't enough data for them to actually see what the viruses looked like, so they had to do a lot more theory. Whereas I think today it seems to be driven a lot more by data, so the theory isn't playing such a big role. But yeah, mathematicians and that are interested in this as well. So I'm not sure exactly what the answer to your question is, if it had a big an effect, but I haven't seen anything.
Yeah,
But I
maybe it's
haven't
just a
also
one-off.
been following it as closely anymore either, since this was research I did in the past. But
Yeah, it may have just been a one-off that perhaps rear its head in 20 years' time or something. Who knows? But I found it
yeah,
interesting at the time.
I mean, there was a virus called SV40 and polyoma viruses, which actually violated the rules of Caspar-Klug. Um, and this was interesting to people too. Like the pictures I showed you had groups of 5 and groups of 6 together.
Yeah.
Polyomavirus is only built out of groups of 5, which would look like that's impossible on this framework, but it sort of followed from the fact that the virus subunits were a lot more flexible than people thought, and proteins are not like little bricks. They're rather, yeah, they're a lot more dynamic, and they can move within themselves. And yeah, so that was interesting too.
Yeah.
Actually, that came out of Caspar's lab about 1980 or so, I think, if I remember the dates right.
It
But
seems
the thing with the Penrose tiling, right? And Graham, you know a lot more about this than I do, is it's almost like there's that quasi equivalence thing going on, right? Where there's a regularity and an irregularity at the same time in the pattern. Do I have that? Is that a misprison?
yes, yes, the Penrose tiling fits over the icosahedron, but it's not in a different way. It doesn't match if you have an icosahedron, a high frequency icosahedron with many triangles, the Penrose tiling doesn't fit over equivalently. One tile matches one triangle. It's quite different than that. So, and geometry often rears its head, and then people say, "Oh, that's interesting," and then go on to other things, and it gets left behind, only to reappear a bit later as something significant. So I just wondered. Thank you.
Thank you, Steve. What I can do is spotlight you for a second. I'll
Well,
get
yeah,
off the
let me get the
spotlight.
blur off this thing, and
Yeah,
I'll,
get your jitterbug.
yeah.
I'm gonna make it so we can see Steve as big as possible so that you can see this model, Dr. Morgan.
Oh, wow.
All right, this one expands out and even can reverse itself and even change shape and sort of breathes at the same. So it's a three-dimensional one of a icosahedron here and expanding out and inverting itself and even changing shape into another structure. Oh, you can see that.
Yeah. Now that looks great. Yeah. And
So you're doing this with the virus, this is a icosahedron sort of, bringing it up to a maximum size and dropping down the other direction, but also has other possibilities in it. So it changes into this kind of thing too. So you can do that.
right.
So, I mean, but it's still built on the same concept of, an icosahedron
Right.
as so.
Yeah, and I know that when I was working in the lab, there was a virus called Hong Kong 97. And it did have like different stages in its development where it would change its shape and it would expand and presumably it was doing something similar to what your structure is doing in there.
There it is.
The capsomeres would twist and yeah. It looks like Bonnie's got her hand up.
Oh, Bonnie, can you come on camera or?
Yes, I'm not all ready for a meeting, but sure,
Well, you
I'll
don't
do
have to.
that.
That's fine. No, you don't
There
have
we
to.
go. Yeah.
Okay.
Okay.
I just I'm gonna spotlight you. Thank you.
Thank you, Dr. Morgan. Your work is so amazing, and it really impacted my work in a significant way many a year ago. But because I've worked with so many of Bucky's colleagues for so many years, prior to many of them have passed. But Don Ingber, I wanted to ask you about his work with the cytoskeleton, the tensegrity aspect of cytoskeleton, and also microtubules, and that structure, that helical structure with the hollow central, it's hollow in the center, that Stuart Hameroff and Penrose had written about as, having so many new kinds of properties. And so I wondered if you have any kind of parallel thing with viruses and cytoskeleton microtubules.
Yeah, I'm not sure if I've got anything too enlightening to say. I mean, I'm aware of Ingvar's root, and it looks really interesting. And obviously, this is the way you want to expand some of these ideas and make them dynamic and look at other biological structures. I can't tell how well received some of this is in the biological community. Clearly, there's bonds between molecules and things that govern how they self-assemble. So it seems to me it's a really interesting, provocative thing. I'm not really an expert in contemporary biology. I'm more an historian, so I'm reluctant to say anything too definitive. But even the idea that we've got to look at the bonds as opposed to the structures as well, that explain how something can self-assemble or how something can move, I think is a key idea. But what do you think? Can I bounce it back to you?
Well, I'm actually writing about it right now. No, I think that, the Science of Consciousness conference happens every year. It's been going on for 26 years, I believe. Last time it was in Spain. This year it's coming up very soon. In any case, that's where you have a lot of scientists. And Don Ingber, I don't think he spoke there, but I believe there's a lot of links between the helical structure of, microtubules and clathrin, which is the most prevalent protein in the brain, as you know. It also has geodesic shape made of triskelions that kind of self-assemble, and that's for endocytosis. So there are transport mechanisms for nutrition and for information. And so I think that especially looking at Raden Twarak's work, and I would highly recommend anybody just see some of her lectures on YouTube. She is absolutely brilliant with the latest applying four-dimensional or quasi-periodic tilings on the virus structure, as well as geodesic shape, as well as, of course, the helical shapes that are part of the virus structure on the inside. So there's all these links between that I believe very relevant. And Don Ingber's lab, the Weiss lab at Harvard, is doing a lot of state-of-the-art research on this.
Yeah, I think that one of the key ideas, it seems to me, is this, which actually emerges with Don Caspar, is this notion of self-assembly, and that it actually, can easily be generalized beyond viruses, and it's probably a really key idea in all of these biological structures, that you need to have a way of assembling them and also disassembling them too, right? And the thermodynamics of how that happens is something that's a key part of biophysics. Yeah, so that, in a way, I think that tensegrity structures have something to say about self-assembly too, right? Like, since they're minimum energy structures, that's what you need to have the self-assembly happen, and it's sort of thermodynamically driven. And presumably that's what happens in clathrin and these other cases too, right?
Absolutely. Yes. And that's what's so beautiful about it, is it's one of the best examples of self-assembly, because it's happening all the time with thousands and thousands inside your body, and especially in the brain. And so they're moving things. And also there's some later research on microtubules and Alzheimer's disease, how they unassemble and then they reassemble. So there's all kinds of dynamics that are being studied now that I think are really exciting, that have a lot to do with that unification between the helical structure and the geodesic structure, the closed system and open system is what I call it. So
there's one interesting case of the virus assembly that goes awry, that instead of forming a closed shell, it forms like a spiral, and it gets bigger and bigger. And so sometimes I guess the self-assembly doesn't assemble the right way. And so viruses can form these strange structures, which is sort of maybe a bridge between having a closed system and an open system too. A lot of the work that Casper and Klug were doing was trying to work out if they could make the subunit have properties so that it always assembled into the same size shape. Because if you're a virus, you want to be, say, a T equals 7 structure. You don't want to sometimes assemble into T equals 3 or T equals 12. And how that happens is what they were sort of interested in. They thought that was built into the subunit itself. I think there's other mechanisms for that can happen. There's sort of scaffolds and things too that may be directed. So it's a more complicated picture.
Well, since it's connected though to the cytoskeleton, which is a tensegrity, you've got that dynamical system in a way. In biology, the biological system has a certain dynamism, and it's expressed beautifully in this stretchy and changeable quality of the cytoskeleton and all of the layers. So, and virus research, your work is so important because I think we forget how important that history is in terms of the discoveries that are made. And I thank you so much for bringing Rosalind Franklin in, because her name has just it's been revived, thankfully, but it's very sad that she left too soon to really get credit for what she contributed.
Yeah, no, I think, well, obviously the public's perception of her is shaped by the Double Helix book, right? That sort of painted her in a certain way as Rosie, the person that couldn't work with other people. And but if you look at all the correspondence, you can see that actually the relationship between Crick, Watson, and Franklin is a little bit more complicated. And they're both friends and collaborators and competitors sort of at the same time. Yeah, and it is tragic that she died and sort of cut off that career. But I think she's been rehabilitated in the 21st century, it seems to me.
Absolutely. Yeah. The book, Dark Lady of DNA, and a number of books are finally giving her the honors that she deserves and should have had back then. But thank you so much for your lecture. I really appreciate listening to this.
Thanks, today. Bonnie, for the kind words.
Thanks, Bonnie. It's great to have you here.
Oh, there's something in the chat.
Yeah, we've got, okay, we're going to bring up Susan. I'm going to bring you up in just a second. I see your hand. Dr. Morgan, can you talk a little about serendipity and science? I mean, I'm thinking about the story of Aaron Klug on the staircase.
Right. Yeah. So Aaron Klug, who's probably one of the most important British biologists of the last 100 years, right? He was president of the Royal Society. His career really only happened because of serendipity in a way. He met Rosalind Franklin on the staircase of the laboratory. It was I think a kind of a house turned into a laboratory on Torrington Road. And he saw her, the same picture I showed you in that PowerPoint, where she had these amazing X-ray photographs, which had a lot of information. You could see all the dots, and there was still this issue about how to interpret what was going on, partly because TMV doesn't form true crystals. It forms paracrystalline fibers. And so had he not run into her when she was holding these things, he would have been studying something else and presumably would not have had the career that he did. So yeah, I think there's no way of reducing serendipity and luck out of our narratives here, right?
I mean, I'm picturing you in the bookstore finding this book.
Oh yeah.
I didn't even know this subject existed. And Aaron Klug on the staircase looking at, bumping into Rosalind Franklin, looking at these images and saying, whoa, 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 whoa. That's what I want to do. I mean, how often does that happen? It seems to be, I'm thinking, it seems like it's in at least one other place in this whole story. But anyway, I just think it's interesting.
Yeah, I've actually got. Well, let me see. I've got this is my current project. I just finished. This is a book on the history of tumor virology, which looks at the way in which viruses and cancer are related. And it turns out that viruses, I'm obviously a little biased here, but I think they're the most important organism of the last 100 years because we study them and we learn about viruses, but then we learn about all these other things that they apply to and more complicated systems. And I think that is true for tensegrity, and it's also true for oncogenes, which is another story. But yeah,
Okay.
even choice to study viruses in the first part, I think, was a lucky serendipitous, or maybe it was more than serendipity in that case, because people believe that you should study a simple system first in order to understand something more complicated.
And look how simple it isn't. Susan, Koenig, just give me another second because I that 1962 paper, Dr. Morgan, if I'm remembering correctly, introduced the term self-assembly, which is a term we end up in biotensegrity using a lot.
Right.
And if I'm remembering this right, there's a point where they say something like, people see viruses as a very primitive kind of structure, but actually there's a sophistication in it. It must have come out of a sort of more full living structure, and now it's cleverly using other life forms, you know what I mean, to do its bidding. Do you know what I'm saying? It's so that a virus can be more efficient. It doesn't have to be an organism. It can be, or whatever we think of it as, a semi-organism or whatever, but it can be a more minimal organism because it's kind of, farming the job of being a full organism out to others.
Yeah.
You know what I'm talking about?
Yeah, exactly. No, I think if you look at this historically, people thought that perhaps because viruses looked more primitive, that they were like in the history of life, they came before cells or something like that. And I think there's some people that still think that, but the dominant view I take it now is that cells came first, and then viruses are like little pieces of cells that sort of almost escaped, like little genes that worked out how to escape from a cell and transport themselves to others. Yeah, so they are. And the other thing to think about here is there's two forces at play. There's the thermodynamics of assembly, and there's also natural selection, which viruses, I think, are the most highly selected organisms on the planet, and that their life cycles are very fast. There's billions and billions of them produced every infection, and only some of those succeed. So because of that, they're incredibly efficient and incredibly well designed to do what they do. Maybe better designed than we are, right?
That's great. Susan, can you unmute and we'll put it over to you? And
Sure.
Jason, I see you've put a couple questions in the chat. Let us know if you can come up to ask them. Go ahead, Susan.
Oh, thank you. That was sort of beyond me, but at the same time, I feel like I took in what I could take in. It was wonderful. I'm interested in maybe having you come a little bit out of your comfort zone and speculate a little bit about origin of life. And I didn't know that. I thought viruses came before cells. That was new information for me. But in terms of demarcations where there's not life, then there's life, there's cells and or viruses and self-assembly, and maybe time frames. Can you speculate a little bit, or do you have thoughts, or you and your colleagues, about how this all might relate to origin of life stuff?
Well, self-assembly is going to be a key idea at the beginning of life, right?
Yeah.
I think that's not too speculative to say. So whatever was in the earliest stages, it has to be something that can reproduce itself somehow. But I
And
think,
I'm sorry.
I think a lot of biologists think that RNA is the molecule that can do all the things they want it to do. And so there's a talk of the RNA world, they call it, which is a time before cells where there'll be RNA molecules doing various things. And then DNA comes later. I don't think there's any definitive consensus on any of this because, it's hard to know. We don't have any fossils, for example, that would show us or, so I'm not sure if that's what you're thinking about or.
It is. Just wondering your speculation. Do you think this issue of self-assembly had to begin or not in a chemical self-assembly?
Yeah.
Versus something else.
I mean, there's philosophers of biology that talk about whether the membranes and the cells came first or whether the replicators inside came first, and whether or not current life is some sort of union between replicating membranes, because any cell is encased with a membrane, and it can reproduce itself too. So somehow you need to get both replicating cells with the membranes that enclose material and also something that is like genetic material that can do things on the inside. I don't have any strong thoughts about what came first or whether or not you get something that's cellular that doesn't have genetic material in it, that's just a replicating like balls of chemicals or something, or whether or not you get longer chain things like DNA and RNA that are replicating by themselves that then later get encased in cells. I don't have any strong thoughts about which is a better explanation.
Do you have any thoughts about when does geodesics show up? When does shape and form start showing up?
I think pretty early. I think that one of the insights from this whole history is that geodesic shapes look complicated, but they're not, because they can be built out of subunits with very simple assembly rules. So the output of the assembly process looks very difficult, but it looks elegant and sophisticated, but if the process to get there isn't, then these can happen relatively early. So spirals and helices and enclosed shapes with like little geodesic domes, I think they can happen pretty early because they are basically chemical.
And one other aspect, and this I may have this out of naivete, but when the virus is not yet alive, what is there? Is there shape? Are there geodesics in the? I don't know if this is even correct, in the non-life virus versus when the virus starts coming alive. Am I right? Inside.
Yeah. So,
yeah.
I think it varies from virus to virus, but some viruses will build the shell first and then they'll put the genetic material inside it somehow. Or they'll build a shell around something that's got genetic material in it. So I think it varies from virus to virus how they do that. But some of the viruses that like that Klug wanted to study, they form shells by themselves just naturally. So sometimes they create real viruses, and sometimes they just have the protein shells. Actually, it turns out that hepatitis B virus does this. And this is the basis for the hepatitis B vaccine, that you make the vaccine just out of the protein shell. It doesn't have any of the genetics involved in it, which is sort of an elegant way of making a vaccine.
Interesting. All right. Thank you, Susan. I'm going to pass it over to you, Jason.
Dr. Morgan, hello.
Hi,
Thank
Jason.
you for your work, especially the 4 papers. The excavation of that history is amazing.
I like
my questions. My questions are in the chat. So it's the energy that Casper's talking about is bonding energy. and that's not exactly the same as the tensegral energy or force distribution. So is Caspar saying that structure is the energy landscape, not the material or the information? And do you ever go into that in your thesis, which I'm waiting on ProQuest to deliver?
I don't, unfortunately. And I don't know the answer to that question, actually, whether or not he thinks the energy is part of the structure or not. I can see it being argued both ways. I think he would say, of course, you need to understand the energy in order to understand the overall structure and its dynamics. I found Caspar himself to be this very, what's the word? Promiscuous with his ideas. Is that a good way of putting it? Like, he had so many ideas when you speak to him, he can talk endlessly about all sorts of things. I think Buckminster Fuller sounded like that as well. And so prolific, yeah, maybe that's a better word. So I'm not sure his later work, like he continued to think about these things, and he sort of was reluctant to publish it, and that's why there was this huge gap between 1962 and 1980. I think he might have liked talking more than he liked writing as well.
Have you read that unpublished manuscript that he refers to?
A long time ago. I think unfortunately it was about 20 years ago.
So you don't have a copy of it that you could shoot over? No. All right. The aspects of and form essays, do those questions persist in your overview of the field, in terms of atomism and morphology and, information in today?
Yeah, I think they certainly persist in philosophy of biology, which is where I'm mostly working. The role of information has sort of been an ongoing topic for philosophers of biology, whether or not it's an essential way of describing life or not. Or, some historians have argued that our focus on information is sort of a byproduct of the fact that molecular biology emerges in the 1950s with the same with computers and Claude Shannon's work on information and all that stuff was in sort of the culture, in the scientific culture, and whether or not there's a different way of describing life that doesn't use information theory terms. Like the central dogma of molecular biology is sort of it uses information theoretic terms. Does it have to, I guess, would be the question. And some people argue that it doesn't. It's just, you could do it more structurally as well.
So do you think virology lost something by Caspar's decision not to, in 1962 to punt on the tensegral framing and just leave it as geometric and,
I think part of it was it probably did, actually, if you think about it, because it's hard to know what that counterfactual is like, right? Had they published something earlier, what science would have flowed from that? Maybe things we don't even know would have gone in slightly different directions. My feeling was that Caspar was wanting to add more and more material to the paper, and Klug was the one that was kind of like trying to keep it more contained. Because I saw drafts, I think he mentioned the Jeremy Norman papers, that there's lots of drafts of papers and things and like what got cut out and what they had, and they changed their mind on various things. And some of it was driven by electron microscopists who were publishing things on a virus structure they didn't like, and so they figured they had to get out there. And there was competition between the people working in crystallography and the people working in electron microscopy, which was also just emerging in the late 1950s as well. So I'm not sure how to answer that question. It would be an interesting counterfactual history. A lot of historians don't do that. There are some that try to say things like, what would have happened if we had taken a different path at this point in time?
I was curious how you thought of it.
Yeah, the one more thing I would say, I think that because of because biological structure was discovered using X-ray crystallography in the beginning, starting with myoglobin and hemoglobin, and we did have this more static picture of things, right, where you're trying to find the structure first, and then you want to do the dynamics. And I guess that's the way. Maybe it's the easiest to do it, but you could imagine an approach where you have the dynamics right from the beginning, and you're looking at changes rather than static structures.
Dr. Levin, is that how you've thought of things, dynamics first?
Absolutely. I'm into my latest talk about that is standing waves, where you have the energy and the matter sort of coalesce as an event, rather than separate at all. Unless you get the energy, you really don't understand the structure. That is so integral to the part of it, so that they coalesce together. So you have to get it. I would assume that, like electrons and protons and things have negative and positive charges. It's that attraction, repulsion energy that's there, and gravity type of forces that are there, it's that forces have to exist in order for these things to occur. So it's the energy in with the matter at the same time coming together. It is, as I said, it's Frankenstein's monster. Without that energy, you don't get that life form.
Yeah. I think in the history of structural biology, you've got two different traditions too. You've got the stuff that comes out of virology that I gave you, that sort of static structures.
Yeah.
You've also got enzymology, where you've got people that are thinking about enzymes and how they work, and they have to move in some way to catalyze reactions, and so they can be more dynamic. And I think people are at more ease at applying thermodynamics to those situations as well, and trying to understand energies as much as they're understanding structure.
Jason, did we get all your questions?
No, I have so many more, but I don't want to monopolize the tea party. And perhaps I could ask Dr. Morgan if he wouldn't mind me contacting him, emailing some questions.
Sure. Yeah.
All right.
Yeah.
I definitely want to open. I mean, I do have many more questions for him.
You're both part of the same part of the world. You're from Australia, he's from New Zealand, so you're close.
Right.
Yeah. We don't want to talk about the cricket right now. But, I'm also only like 60 minutes down I-95 from you, so I promise not to knock on your door.
Okay.
All right.
Thank you.
Thank you, Jason. We're getting to the end of our time here. There's nobody else with a hand up. I probably could talk for another hour and ask questions. Dr. Morgan, you've been really
Can we
generous.
do that?
You've been really generous with your time. Would you be willing to do well, let me ask before we do closing toast. Did you have anything to add, Steve or Graham, before we close up?
A quick 2 minutes.
Yes, take your time.
Just a quick moment on your new book coming out. Tell us a bit more about it in a few sentences.
I think it's been out for a few years, right?
Yeah, it's been out for a couple of years. It's at Johns Hopkins University Press. It basically tells a century of research into the idea that viruses cause cancer, starting with people that were kind of seen as outsiders and ending with the HPV vaccine, the hepatitis B vaccine, the discovery of oncogenes, and along the way, a test for the AIDS virus. And so it's a case, I think, led to about 7 different Nobel Prizes. And so it was a very productive model organism to focus on. And I really enjoyed writing it and interviewing a lot of these eminent scientists, many of whom have died in the last few years, unfortunately. But yeah, I think it's one of the more interesting strands in the history of 20th century biology, and it's just not generally known. So that was one of my motivations for the book.
Oh, thank you.
I have a question too. I guess I shouldn't be asking it, but what's your current?
Wait, hold on a
passion?
sec. I want to make sure, are people able to turn their videos on, or are they not able to? Let us know in the chat if you
There's
can't,
Carol.
because Carol,
Maybe-
Disabled.
Oh,
did you have a question as well, Carol, or just waving? Okay. That it's okay that you can turn your video on. Okay, great. Go ahead, Chris.
I'm just curious to know, what's your current passion? What are you working on or excited about right now?
I'm what am I doing right now? I'm kind of at a I'm looking for another large project. I've got little things that I'm doing. I've got a little project on the philosophy of collecting.
Hmm.
I'm a collector in my spare time, so I'm kind of interested in like the rationality of that. Yeah, there's a few little projects. I'm sort of looking around for another big one. Like the book itself, it took 10 years to write. So picking the right thing to do is sort of important. And unfortunately, I've been pulled into university politics and administration a little bit. So I'm chair of the faculty senate right now, and that's taking up a lot of my time.
Very time consuming. So does it happen as you wait until something really calls you? And that's what carries you down the path?
Yeah, I mean, I think there's different models. Some people like working in big groups. I actually like finding a project that no one else is doing. So, and I can be sort of one of the first people to think about something, and then there's less pressure that someone's going to scoop you or like publish before you can. So that's the sort of thing I like.
Well, try putting Biotensegrity on that list.
Oh, maybe I should. Yeah, that's a good suggestion. Yeah, no, that would be an interesting one. The history of biointegrity, although I think that all the experts are maybe here.
Right.
All right. And that idea of getting scooped, that's also in your papers of that time, that idea that other people were also looking at geodesic domes.
Right. Yeah, I think.
And Casper and Klug were like, well, we better get our paper out because.
Yeah, this is, I think, one of the ways we try and fine tune science, because you've got to get the right balance between cooperation and competition. And if it goes too far in one direction, things kind of slow down. Jim Watson, in fact, he liked a lot of competition. He thought that's the way you drove it forward. But I'm more of the view that you need both, and how to balance the two is a really interesting problem that deals with basically the social structure of science, how we organize as a social group.
Right.
Yeah.
All right. Well, Dr. Morgan, I'm going to turn it over to you for our closing toast.
Okay. Okay, let's see. What can I toast? How about I toast to continued interaction among different disciplines, interdisciplinarity.
Hooray for that.
Cheers.
Hooray for that.
Cheers.
And thank you so much for inviting me to your group. I really enjoyed it.
We're going to